bulk or single cell attack, or vice versa? If so, to what extent and how much abundant information? Bulk or single cell to predict chromatin accessibility, or vice versa, to what extent and how much redundant information. So this is, you know, too common of a project, many attempts, not clear answers. So I would say that it's sort of, you know, not quite doable, but I think that the first one is great. All right, so idea number one, mapping intron length evolution trends on eukaryotic phylogenetic tree. Cool. Investigate codon gain and loss in other eukaryotic lineages. Okay. Creating a model of ocean protist diversity based on ecological conditions. So Tatiana, are you kind of set on I want an evolution project basically? Because none of these like strike me as slam dunk basically. Let's yeah, let's come back to those. So I'll have to think a little bit about them. So Pablo, map the occupancy of chromatin associated proteins in breast cancer cell lines. Occupancy. Perfect. In the map, that's the estimated cell type. Use the data in this paper for breast cancer. Yep. Sounds great. Still missing a question, but big data. Insertion from this paper in attempt to find precision factor binding sites developed TBINet, preparing for conservation methods. We tried to research reflecting human transcription factor footprints. So, yeah, I love it. Great building blocks. Investigated RB Fox motifs, a family of splicing factors that regulate alternative splicing during development. From this study, they found several motifs that together exert regulation of these genes. This paper finds that RNA splicing factors can act as oncoproteins in tumor sequencers. Found motifs in response. Yeah, so I would say that this is a bit too open-ended, you know, and very risky, insufficient data density space. Pablo, do the responses make sense? Okay, Ricard. I'm interested in learning representations of genetic patterns by applying neural language modeling of genetic sequences. Predict the next token SGT from previous tokens. A sequence model such as transformer, contextual embedding for the nucleotides used for downstream tasks. Specifically, I'm thinking that assignment search might be a downstream task. Evaluate the model in downstream task benchmarks. The good thing is that the data for genetic sequence abundant. So I'm not sure I fully understand this. Are you basically sort of looking at haplotypes or, oh, by the way, is my, oh yeah, my sound is Oh, I think the problem is like my connection is pretty horrible. Can you, Daman, can you guys hear me? Okay, so Ricard, it might be your connection. Yeah, I cannot hear you. I'm sorry.
But
oh,
you can maybe you can. So, I mean, what I'm trying to say is to just like. Do you hear me now, Professor?
Yeah, now I can hear you. Yeah.
Okay, cool. No, so I'm just thinking that like genetic sequences are more abundant than labeled data. So you would, and this is pretty common in NLP where you pre-train your models on like you scrape Wikipedia or something, and you learn contextual embeddings of words and sentences, and you could do the same thing for genetic sequences, just like run these models on a lot of genetic sequences to learn representation of genes, like contextual representation, and then you can use those for like alignment and search, etc., or like even other downstream tasks.
So I love the area, but I'm not sure that the context makes sense. So what do you mean by context? Basically, what I'm concerned about is that genetic patterns. When you say genetic, do you mean genomic or genetic?
Don't know the difference, but I mean, I just need like.
Let me explain. So genetic means inter-individual differences, whereas genomic means just the DNA sequence. Do you mean genomic?
Yeah, I mean nucleotides, just like that's what I token, just like put up in a long sequence.
So good. So I love the area, and what I'm thinking about this is that working at a higher level makes more sense. So use motifs as your tokens rather than individual nucleotides. You see what I'm getting at?
Yeah, great feedback. Thank you.
Okay, perfect. I love it. I think this is just a great suggestion. So, I think you should definitely do that problem.
Thank you.
The topological
Thank you.
analysis, prediction, protein shape. Yeah, complete information topology. So I would say that protein folding is way too hard for a term. Modeling proteins as graphs and then understand the function from the amino acids of the graph by learning theory perspective. These are graphs, okay, amino acids, learn isomorphisms. So I would sort of focus on graph isomorphisms on different areas of biology, e.g. regulatory networks or networks of cell-to-cell similarity in single-cell data, etc. I think that the area of graph isomorphisms is totally worth it, but I feel that the specific application of protein folding is probably too hard. Does that make sense?
Definitely. Thank you so much.
All right, Jessica. Applying machine learning to RNA sequences for intrinsic subtyping of specific cancer types. Paper PAM50 50 breast cancer cell with highest concentration of the 5 subtypes centroid. Immune strategy, average within class. Improve consistency or reproducibility of PAM50 subtyping by averaging within class mean. Multi-class reduce the regression to the robust single sample classified strength and breast cancer subtyping. Expand on their research and new project to stratify other diseases while iterating on RNA sequences from tracing. Tu peux fermer ma porte, mon trésor? Non. Moi, je suis descendu chercher le mien. Strategy. When you say RNA sequences, is this about RNA folding or do you mean mRNA sequences? Jessica, can you help me out here?
I think it's mRNA sequences.
Okay, but not about folding. So you're
but not about folding.
Okay. Intrinsic subtyping of specific cancer type. Do you mean expression levels?
Yeah.
Ah, got it. Thank you. Okay, perfect. Okay, good. Now everything makes much more sense. Okay, the expression of the 50 cancer genes. Great. I love it. Multi-class logistic regression, robust single cell. Stratify the disease by other diseases. Okay, so yeah, iterating on their strategy. Sure. So yes, mostly reusing algorithm on new data. The novelty is not super high, but and then the challenge might be pre-processing, find the right data sets, sufficiently homogeneous diseases, and sufficiently heterogeneous for. So I kind of like it, but I think you need the killer application. Basically, I think it's it's cool, and I think my recommendation would be basically number one, find killer data set, and then number two, modify algorithm, to match it somehow. So basically, I think your project will greatly strengthen from, being able to actually have some new computational novelty as well. Does that make sense, Jessica?
Yeah, thank you.
Great. In this paper I looked at last week, researchers developed a shallow HCNN of LungNet, analyzes CT images, predictable survival, non-small lung cancer. LungNet successfully predicted survival outcomes for 4 independent cohorts and 4 medical centers, 65 patients with high risk, low risk. Transfer learning frameworks on pre-training model. And LungNet itself is not a direct application to genomic data. The concept of predicting survival outcomes includes transfer learning. Yeah, I love transfer learning. It's a great idea. Cancer genomics, I like that. These data sets exist that correlate expression of disease markers with prediction outcomes after training the model survival prediction. Transfer learning. Yeah, I like that. I like that. So basically, number one, use transfer learning to find relevant features, and number two, apply these features, prioritize features for a new task. Challenge is going to be to basically the distance, find the right distance between training and application. If you have sort of the killer data set again, it'll be, nice. Does that make sense, Jessica?
Yeah.
Okay, idea 3, looking at how gene expression in cancer. And by the way, we're gonna wait. We're gonna go way over. So let me see. Oh shoot, I'm supposed to be at a discussion with an NIH officer. So I really like having you guys live as I'm giving you feedback. I don't know what the best way to do that would be, but what do you guys think? Should we just do another session on Monday at 4 p.m. where all we do is just go through the proposals? And then what we could do is sort of have people show up in sort of roughly 3-minute intervals based on your expectation for when I'm going to be going through your ideas. But these are really awesome ideas. It's just that as I'm going through them, this interaction, however low throughput it might be, is sort of low latency, which is nice. So okay, so let me do a poll, I guess. So let's see. A, yes, more feedback live would be great. B, now just enter your comments at the end. And then C, okay with meeting for a while on Monday altogether. D, staggering teams to, or staggering students to roughly time. And then E, you could Skype each of us comes. All right, so let's do the poll here. So results, and then oh my God, I've never seen a poll so flat. So it's 12, 12, 10, 10, 14. It's hard to choose. Okay, each of us when our turn comes, or yes, more feedback live would be great, or 9 to your comments at the end, or okay. Okay, so here's what I'm going to try. I'm going to try now redoing the poll with only one option possible for each person. So let's retry that. Your answer. Okay, so it seems that option B is winning out, and E to some extent.
I see a sign of our oh my goodness, one of my favorite.
Hey guys, okay, so it seems that the answers are now 4, 8, 2, 3, 9. So B and D. So now just enter your comments at the end and Skype each of us. Okay, perfect. So I'm just gonna do it offline, and if I have questions, I'll just enter comments directly on the Google Doc. Okay. So TAs, let's do some breakouts and then have people chat. Does that sound like a good plan? Bye guys. Sorry for all the connection troubles today. Bye.